Distinguished ladies and gentlemen, please, we want you to be aware that in the next couple of minutes, the academic procession will be coming in. So we want you to please take your position, take your place, make yourself comfortable. Any moment from now, the academic procession will file in. Please, shall we now rise for the academic procession? Thank you.
Deputy Vice Chancellor Administration, Deputy Vice Chancellor Academic, Registrar, University Librarian, Provost of the College of Medicine, Provost of the Postgraduate College, the Dean of the Faculty of Pharmacy, Deans of other faculties and of students, Directors of institutes and centers, Heads of department, the 12th Vice Chancellor of this university, distinguished guests, ladies and gentlemen. The 567th University of Ibadan Inaugural Lecture will be delivered this evening by Professor Iruka Okeke of the Department of Pharmaceutical Microbiology on behalf of the Faculty of Pharmacy. Professor Iruka Okeke was born the second of 4 children to Engineer Obed and Mrs. Flora Okeke in 1970. She attended Queen's College, Lagos from 1980 to 1985, where she won the Ordinary Level Biology Prize. She attended Obafemi Awolowo University, Ile-Ife, where she obtained a Bachelor of Pharmacy degree in 1989, topping her class and winning 5 graduation awards. She did a pharmacy internship at the Obafemi Awolowo University and now registered by the then Pharmacists Board of Nigeria, now cancelled. She did the mandatory National Youth Service Program in 1990 at the then General Hospital Ikeja, now Lagos State University Teaching Hospital, between 1990 and 1991. Professor Okeke was appointed Junior Training Fellow at the Obafemi Awolowo University in 1992 and earned an MSc in Pharmaceutics, Microbiology specialty in 1994. She received a PhD from the same institution for the work done at Obafemi Awolowo University, Ile-Ife, and the Center for Vaccine Development, University of Maryland in the United States, which was supported by the Fulbright Fellowship. A doctoral thesis received the 2001 Nigerian University Postgraduate Thesis Award in Science category. Upon the completion of her PhD and a 3-month International Federation of Science Research Fellowship at Uppsala University in Sweden, she returned to Obafemi Awolowo University, Ile-Ife, where she taught briefly. Professor Okeke then proceeded to the Department of Microbiology at the University of Maryland Medical School for postdoctoral training in molecular microbiology in 1998. In the year 2000, Professor Okeke accepted a career development lectureship at the University of Bradford in the United Kingdom. She was appointed assistant professor at to the faculty at Haverford College, a research active undergraduate college in the United States, in 2002. During her time with the biology department at Haverford, Professor Okeke received 3 consecutive grants from the National Science Foundation and other intra and extra moral awards. She spent her 2005 sabbatical leave at Wellcome Trust Sanger Institute, Cambridge, as a Branco Vice Fellow at the Society in Science Switzerland from 2005 to 2009, and was a fellow of the Wissenschaftenkolleg zu Berlin from the year 2010 to 2011. Professor Okeke received tenure and was promoted to Associate Professor of Biology and Adjunct Professor of African Studies at Haverford in 2008 and to Full Professor in 2014. She also received the Pharmacy Ambassador in the Diaspora Award for her alma mater at the Faculty of Pharmacy at Obafemi Awolowo University. Also in 2014, Professor Okeke received the African Research Leaders Award from the United Kingdom Medical Research Council. She used this to relocate to the University of Ibadan, where she set up a molecular biology research laboratory in the Department of Pharmaceutical Microbiology in the Faculty of Pharmacy. She became an adjunct professor in the Department of Medical Microbiology and Parasitology, College of Medicine in 2023. From the University of Ibadan, Professor Okeke works with the Nigerian Center for Disease Control and Prevention, NCDC, to develop and implement antimicrobial resistant policy. Professor Okeke was the second editor-in-chief of the African Journal of Laboratory Medicine, taking over from John Nkengasong in 2017 and completing her second term in 2022. She is currently a senior editor of Microbial Genomics and has previously served as editor for the Journal of Infection in Developing Countries, Frontiers in Microbiology, Lancet Infectious Diseases, and PLOS AMR channel. She was co-lead of the scientific committee for the Welcome Connecting Science Antimicrobial Resistance and Big Data Conference from 2018 to 2021, and was also co-chair of the scientific committee of the African Society for Laboratory Medicines Conference in 2023. Professor Okeke's research and training initiatives at the University of Ibadan have been supported by competitive extramural awards from the National Science Foundation, among several others. Professor Okeke serves and has served as advisory board and panels of the Professor Okeke is the author of scholarly book, Diving Without Seeds, The Case for Strengthening Laboratory Medicine in Africa, that is published by the Cornell University Press in 2011, co-author of the textbook, Genetics, Genes, Genomes, and Evolution, published by the Oxford University Press in 2017, and co-editor of the Antimicrobial Resistance in Developing Countries, that is published by Springer in 2009. Professor Okeke has authored or co-authored 14 contributions and other books and 180 journal articles. Collectively, these works have recorded almost 12,000 citations. Professor Okeke was commissioner in Lancet Commission for Nigeria 2022 and led the first paper in the Lancet Antimicrobial Resistance Series, which was launched at the 2024 World Health Assembly, briefed the 2024 United Nations General Assembly, and was featured in Nature in September 2024. A teacher, scholar, Professor Okeke has mentored over 120 research students, the majority of whom continue to work in science and health. Professor Iruka Okeke is a member of the Guild of Ushers at the Chapel He's a member of the Guild of Ushers, Chapel of the Resurrection, University of Ibadan. Professor Okeke is happily married to a writer and human rights activist, Dr. Ike Okonta, in 2012, and they have a daughter, Chika Ifeyinwa. Distinguished ladies and gentlemen, I now invite Professor Iruka Okeke to present a lecture which is titled Genomic Revelations, Messages in the Genetic Material of Our Bacteria Adversaries. Professor Okeke.
The Vice-Chancellor, Deputy Vice-Chancellor Administration, Deputy Vice-Chancellor Academic, Deputy Vice-Chancellor Research, Innovation and Strategic Partnerships, the Registrar, Librarian, Provost of the College of Medicine and the Postgraduate College, Dean of the Faculty of Pharmacy, Deans of other faculties and of students, Directors of institutes, distinguished ladies and gentlemen. In this 567th University of Ibadan Inaugural Lecture, I'll present my group's research findings and their significance as gleaned from the genetic material of bacterial adversaries. I'm a microbial geneticist who turned to genomics 2 decades ago, returning to Nigeria after 17 years of work abroad. While inaugural lectures typically encapsulate a scholar's life work, I will instead use this lecture to emphasize the most recent third of my research journey. Essentially, I'm devoting this lecture to the amazing discoveries that my students and I have made at this great institution, appropriate for the 17th inaugural lecture in the Faculty of Pharmacy, the 4th in the Department of Pharmaceutical, Microbiology and Biotechnology. I'm honored to be the first since our department was given its new, extended, and more appropriate name. I thank God for making it possible for me to stand before you to share work that He enables and inspires, and the Dean of the Faculty of Pharmacy for inviting me to do so. A microorganism, or microbe, is a living being that cannot be seen without a microscope. Previous inaugural lectures from my department have all at least touched on the battle against pathogens, that is, disease-causing microbes, which is a significant preoccupation of pharmaceutical microbiologists, alongside studying microbes that alter medicines. Indeed, look at the titles of microbiologist lectures in pharmacy, medicine, veterinary sciences, and the ongoing conflict between humans and microbes that can harm us comes to the fore. Bacteria, my favorite microbes, are everywhere, on our bodies, in the environment, even in niches where we cannot survive, such as the ocean floor, or mountain tops, or hot springs. Everything that you, I, and all living organisms, including the aforementioned bacteria, are equipped to accomplish is written in our genomes, often called life's blueprint. Each cell's genome is made up of DNA strands tightly wound and stuffed into its nucleus, if it had one, or for bacteria which do not, suspended in its cytoplasm. These threads contain all the information that dictates how the cell is made, how it functions, where it can and can't survive, and what it can and can't harm. Translucent threads, when bunched together, can sometimes be seen, even lifted and spooled during extraction. These threads are strings of 4 nucleotides, which are DNA letters. The lettering itself is submicroscopic, thus DNA is read biochemically rather than physically. I'm often approached by excited students that want to see sequencing in action. But sequencing is boring, and it's automated. As with literary letters, watching a typist or a printer work is mind-numbing for most and unattractive to creative scientists. The excitement lies in reading the information. With this, we can make sense of a bacterial cell without even seeing it, understand its history, build intelligent tactics for battling it. The pen is indeed mightier than the sword. Words in your genome can be used to determine who your parents are, whether those you consider as your children are yours, or which illnesses you might suffer. The code is universal, written in the same language for all creatures, giving those of us who have learned to read it insight into many things, perhaps too many. But fear not for your privacy. I have no interest in your genome. I find bacteria much more fascinating. For me, an E. coli genome is more intriguing than a Homo sapiens one ever will be. In the context of cellular literature, the human genome is an enormous corpus, But the bacterial genome is analogous to a child's fairy tale, containing mystery, excitement, but still being small, compact, and easy to read. Bacterial genes are very close together. Sometimes they overlap. There are no in-between dull scenes, making for a thrilling, fast-paced read. Due to their small size, bacterial genomes are cheaper to sequence and require less computational power, internet bandwidth, and frankly, even electricity to analyze. These are important considerations if you're doing cutting-edge genomics research in Nigeria, as we are today at the University of Ibadan. The African continent is the place to do genomic research because we have more biological diversity than anywhere else on Earth. Much of my group's work is descriptive as we work through to catalog life. However, we always push beyond understanding what to ask how and why. Hypothesis testing in our understudied terrain inevitably leads to discovery, even if it takes a long time. For this reason, I believe my work has been more impactful since coming to Ibadan, but I'll leave you to be the judge. Our group studies bacteria that can cause disease, particularly in very young children. I salute all our health workers who care for children with these infections. It has been a privilege to collaborate with the best of you, and thank you for your heart for the work. Our primary goal is to help generate evidence and tools that will make your job easier. E. coli is my favorite species. For my entire career, I've focused considerable effort, or no, I've been obsessed with E. coli that cause diarrhea. In our country, far too many children are killed and disabled by diarrheal disease. As a doctoral student, I studied bacterial causes of childhood diarrhea in and around Ile-Ife. Our research found that the most common type of E. coli implicated in infantile diarrhea in that context was enteroaggregative E. coli, which remains till today poorly defined. Enteroaggregatives are so neglected that they're not included on official lists of neglected pathogens, and fewer resources are invested in research on them than on canonical neglected diseases. Our enteroaggregative E. coli research has thrown light on the basic science around bacterial colonization. Adherence factors such as those we have discovered can account for colonization, however, they cannot account for diarrhea. Having performed multiple smaller diarrheal studies in Nigeria and Ghana, we set up a large case control study to identify truly virulent enteroaggregative subcategories in 2 northern Ibadan's peri-urban local government areas. Enteroaggregative E. coli as a broad category was not associated with disease. So many well children carried the bacteria. So we had to scratch below the surface and read the genomes of the organisms to figure out the epidemiologic nuances. By carefully reading the genomes of enteroaggregative strains from children with diarrhea and those from healthy children, doctoral students Bisi Akinlabi and Rotimi Dada independently coalesced around a family of secreted proteins known as spates. One spate, Sige, was significantly more commonly found among enteroaggregatives from children with diarrhea than from those without. We've been able to provide a real-world human data to corroborate an earlier report that showed that deleting Sige from an enteroaggregative strain abrogated its virulence in rabbits. In 2017, we initiated a drug discovery project to find the forerunners of medicines for treating neglected bacterial infections. Naturally, we began with enteroaggregative E. coli. We competed for and won access to the chemical library pathogen box, which is provided to innovators in low and middle-income countries that are tackling neglected diseases. This is an example of an opportunity open to my group in Ibadan that we would not have been able to access before I came to this institution. Dr. David Kwasi, my then lab manager, built himself a drug discovery PhD program in pharmaceutical chemistry. He measured the ability of 400 compounds in the pathogen box library to hinder the strains from colonizing plastic, forming a multicellular colonization infrastructure that we call a biofilm. We found 5 such compounds in David's screen, and all of these hits showed greater biofilm inhibition and less growth inhibition than the only known enteroaggregative E. coli biofilm inhibitor. Living cells, even bacterial cells, are complex. Therefore, finding the mechanisms of action can be a needle in a haystack, lifelong endeavor. In our case, however, the task was simplified by exploiting our local interrogative E. coli genomes. When we screened a subset of them against David's 5 hit compounds, strains that possess the aggregative adherence protein AAP gene were susceptible to the activity of the hit 687-800, whereas those lacking this gene were not. And then we confirmed that AAP is a target of 687-800 by testing interrogative E. coli deleted for the AAP gene and found that susceptibility to this compound was lowered, showing that AAP is one of multiple targets for this compound. By now, I hope that by being introduced to one of the most important neglected bacterial pathogens, you're enthralled by keys and cues that can come from reading DNA. The DNA speak that guides our discoveries is not hard to understand, but it does need to be learned. Much of my scholarly career has been devoted to teaching students to read and decipher this language of life. My students' collaborators must receive the appropriate foundation to make intellectual as well as technical contributions to our joint academic endeavors. With former colleagues at Haverford College, where we taught introductory genetics using a primary literature-focused genomics first approach, I co-authored a genetics textbook that solves this problem by teaching students the fundamentals of reading DNA even before they learn of Gregor Mendel's work. The second teaching approach I carried away from Haverford College is delivering practical science instruction through inquiry-based or discovery-based classes, which allow our students to make discoveries as they gain key competence. In the Department of Pharmaceutical, Microbiology, and Biotechnology, we now teach inquiry-based modules on antimicrobial resistance, water quality, and the phylosphere. What is the phylosphere? It's a collection of bacteria that lives in and on the aerial parts of plants. Phylospheres are large collections of friends that, unlike disease-causing pathogens, can be interrogated with little risk. Therefore, we use the phylosphere to formally but safely instruct inexperienced students to do the microbiological, bacterial colonization, molecular biology, and genomic research. In my first year at the University of Ibadan, Dr. Okeke and I co-taught a phylosphere class, and one of her students was hooked. Anderson Oyakena pivoted his master's project towards phylosphere studies and followed this with a PhD that Dr. Koka and I co-supervised with Professor Berg at Graz University in Austria. Together, we characterized the phylospheres of 2 Nigerian medicinal plants for the first time. We bypassed tedious procedures that would have been needed to identify the cultured isolates using first principles Bergian manual schemes by sequencing their 16S ribosomal RNA genes and naming them by comparing the sequences we obtained to a database. Anderson isolated at least 7 novel species in the course of his single PhD project. His and other students' findings illustrate that there are bacteria that we don't know, that know us and know our plants. Who better to discover bacteria that live in Ibadan, but are known to science than University of Ibadan students? In 2016, Dr. Chikwe Ihekwazu was appointed the Chief Executive Officer of the Nigeria Center for Disease Control. My longtime collaborator, Professor Dikpo Aboderin from Obafemi Awolowo University, and I, fresh from having worked with 5 East African countries on their plans, contacted Chikwe right at his appointment to request that he assist in getting momentum on national action planning in Nigeria. Chikwe, whose personal slogan is "Keep Pushing," had the Minister of Health, our own Professor Adewale, to assign the task to his agency and appointed us to coordinate the efforts. Working day and night with a dedicated team from NCDC and academia, we performed a situation analysis, engaged stakeholders, and wrote Nigeria's first national action plan for antimicrobial resistance, which the Ministries of Health, Agriculture, and Environment endorsed. The plan was presented to global surprise at the 2017 World Health Assembly. Nigeria has since built on this effort, and our Minister of Health and Social Care launched a second national action plan against antimicrobial resistance earlier this year. Included in the first plan was a pressing need for Nigeria to set up a nationally owned and quality-assured surveillance system for antimicrobial resistance. Such a surveillance system must have a national coordinating center, which for us is NCDC, at least one Sentinel site, and a national reference lab that offers a suite of quality assurance functions, which, using traditional confirmatory and subtyping methods, requires multiple scientific teams and complex instrumentation to deliver. We made the then radical decision to provide all of these functions using a single method, whole genome sequencing. And so we became the Ibadan node of the Global Health Research Unit for Genomic Surveillance of Antimicrobial Resistance, and we're twinned with UCH's Medical Microbiology Department to perform National Reference Lab services for antimicrobial resistance. Once we sequence the DNA of bacterial isolates for antimicrobial resistance surveillance, we can learn other things about them, further increasing the cost-effectiveness of this approach. Investigators are presently developing Klebsiella vaccines that will be used to prevent newborn sepsis, one of the most devastating healthcare-associated infections. Currently, newborn infections are a major roadblock in the way of meeting the SDGs, and as there are very many different protective antigens for Klebsiella bacteria, we needed to be sure that when we deploy vaccines, they target the right ones. Identifying surface antigens on bacteria is tedious, difficult, and expensive. Expectedly then, until recently, there were few data describing prevalent antigens from Africa and none from Nigeria. But now that we sequence Klebsiella for antimicrobial resistance surveillance, we can extract information about antigens from those genomes at no extra cost. We've now made reports on retrospective isolate sequence and will continue to do so prospectively. Perhaps the most important information type we gain from studying genomes of resistant organisms is an understanding of how closely related they are. Just as my DNA is more similar to my sister's than anyone else's in the room, sibling bacteria have a similar core DNA to each other, their parent, and their descendants. A handful of changes in DNA occur during reproduction, and these copying errors are passed to progeny. Biologists commonly display information about relatedness in simple tree diagrams. An understanding of the error rate and a count of the number of differences between two bacterial genomes makes it possible to compute the degree of interrelatedness. Combining this information with epidemiological data produces the best tracking approach we can presently imagine. In genomic epidemiology, we use sequence to determine whether organisms from different patients or sources are related. This way, we can confirm or rule out a cluster of infections as an outbreak, sometimes even determine how the outbreak began and spread. And so when the Obafemi Awolowo University Teaching Hospital Complex resident, Dr. Emmanuel Irek, explained that he had noticed an uptick in the number of patients colonized with Acinetobacter bacteria in his ICU, in his hospital, we were able to compare the sequences from his ICU isolates with those of Acinetobacter from elsewhere. And then PhD student Dr. Ekesin Odi, who did the comparison, built the tree before you. The isolates from the ICU, starred and designated clade 1, differ by only 2 or fewer genetic changes, suggesting they come from a point source and are descended from a very recent ancestor. This confirms Dr. Irek's suspicion of an outbreak and provided the evidence that he needed to boost infection prevention and control in his hospital. Since then, on request, we have investigated 5 suspected outbreaks of opportunistic pathogen infections in hospital at the request of clinicians. We confirmed 2 of them and we ruled 3 others out. Even more concerning than the possible outbreaks that are brought to our attention by watchful clinicians is that in the course of routine sequencing to support Nigeria's antimicrobial resistance surveillance system, we have uncovered almost 50 hospital outbreaks retrospectively, all of which went unnoticed when they were in play. Only a fraction of Nigerians have access to health facilities. Therefore, both patients and health workers would agree that it is unconscionable that Nigerians can become sick because they visit a health facility. This is unfortunate, but it's a common occurrence worldwide with very high rates in Africa, including Nigeria. In our overcrowded and under-resourced health facilities, it's especially difficult to prevent healthcare-associated infections. And as most patients are never cultured, we frequently don't have the input material for an investigation. More patients need to have access to appropriate lab testing. Diagnostics development will support our health system and preserve patient lives. On our side, identifying outbreaks retrospectively has been commended, but it's not enough. Our prospective attempts, while successful in the end, have been too slow to support deploying interventions in the course of those outbreaks. Our turnaround times are prolonged by procurement difficulties, power cuts, inadequate sentinel data systems, seemingly trivial issues that represent formidable roadblocks to better patient safety. In community outbreaks, which originate outside hospitals, reading germ genomes has also helped us to better understand how harmful organisms spread. The devastating point source COVID-19 pandemic, in which an estimated 7 million individuals lost their lives, is a case in point. It's worth mentioning because it brought the utility of whole genome sequencing for public health surveillance to the attention of non-scientists, and showcased that this is an area that Africans can work from scratch to lead in a little over a year. Shortly after beginning whole genome sequencing of bacterial genomes, we were requested to contribute to the COVID-19 response by sequencing SARS-CoV-2, the virus that causes COVID-19 in Oyo State. As we neither have a virology lab nor the expertise to run one, this task was accomplished in collaboration with the College of Medicine's Biorepository Clinical Virology Lab, which was set up and run so admirably during the pandemic by Dr. Adeola Folotade and her team. Working with them in an effort led from our end by then master's student Elizabeth Akonde, we sequenced hundreds of viruses in the pandemic's Delta and Omicron waves, and were able to demonstrate that those lineages circulated among us, as well as to find variants that were believed to be absent in Nigeria. In addition to variant typing, we supported infection prevention and control efforts to ensure that routine services for patients with typhoid, malaria, and other fevers could continue to be offered at UCH and the College of Medicine's Ibarapa Rural Program. Mr. Vice Chancellor, ladies and gentlemen, we are proud to support NCDC and Oyo State and to be part of what was one of the best public health responses to COVID-19 in the world. However, without complaining, I should let you know that we worked on ending hours all weekends under difficult circumstances, including a strike without pay. COVID-19 first responders who did this sacrificially around the world and did not lose their lives in the process received as their reward ridicule and harassment. For us, it was thankfully more of the former. We had added worries about core work that piled up in a backlog that we're still struggling to address. I thank my team and scientists engaged in the COVID-19 response worldwide for selflessly carrying this extra load. It brought new respect for each of you and what emergency first responders bear more routinely. When not diverted by emergency response, a lot of our community surveillance efforts has been given to invasive salmonella. With the Severe Typhoid in Africa Consortium, in collaboration with the International Vaccine Institute, we began intensive surveillance of typhoid and other infections caused by invasive salmonella. The goal was to compute the disease burdens from Salmonella typhi, the cause of typhoid fever, and non-typhoidal salmonella, and to determine how and where the new typhoid conjugate vaccine should be used. This massive effort involved staff at 4 Ibadan health facilities and investigators in 5 other African countries. Surveillance from 2017 to 2019 showed that the typhoid burden in metropolitan Ibadan is between 29 and 49 cases per 100 person years, and that most of this burden is borne by children aged 2 to 14 years. During the SETA study, management of over 170 intestinal perforation cases, a severe manifestation of typhoid, was overseen by our surgeons at UCH and Our Lady of Apostles Hospital, Oluyoro. Thank you. Perforation rates in Nigeria, DRC, and Burkina Faso were the highest across all study sites, pointing to the very severe manifestation of this dreadful disease in our setting. We sequenced all salmonella isolates from surveillance in Nigeria and confirmed that most of our bloodstream salmonella isolates are indeed typhi, the serovar that causes typhoid. We did indeed find, as seen elsewhere in Nigeria, the Salmonella typhimurium is present in Ibadan, but Salmonella enteritidis was more common. And the most unexpected finding was Salmonella velveridum, a serovar rarely reported from Africa. Now that we have an antimicrobial resistance surveillance system, we can easily determine whether similar or identical pathogens are being isolated elsewhere in our country. And what we found is instructive as well as valuable. The typhi and enteritidis lineages that predominate in Ibadan are circulating elsewhere in the country. In this regard, most of the learnings from the SETA study have national relevance. But the detection of valveridin does not. We did not find this cerevar anywhere else. Interestingly, we have recovered valveridin from stool specimens and household water in Ibadan. Thus, valveridin is, at least for now, uniquely in Ibadan. Writers Chinua Achebe and Chimamanda Adichie warn of the dangers of a single story. Our finding of a local endemic focus for Salmonella valveridin showcases the perils of extrapolating from one study or one city to inform a whole country or continent's health policies, as is common in Africa. We must underscore the importance of geographic representation and the need to build global surveillance information from locally derived data. This can be done properly, ground up with local ownership, as we do in our antimicrobial resistance surveillance system. Nigerian cities, including Ibadan, are increasingly besieged by floodwater and paradoxically live in a perennial struggle to access potable water, in a burden borne quite literally on the heads of Nigerian women. I therefore read with interest Oluyiton's History of Sanitation in Ibadan. The take-home message from the chapter on water is that insufficient investment and outright neglect mean that most of us cannot expect to get water, let alone clean water, when we turn on our taps. With Dr. Alabi in my department and our students, we leveraged the SETA healthcare utilization surveys to collect water samples from 250 households each time in Ibadan and analyzed each sample for microbiological quality. The results are scientifically interesting and socially alarming. All of our surveys indicate that less than 20% of household water in and around Ibadan is potable, that is safe for drinking. Almost all Ibadan residents get their water directly or indirectly from a groundwater source, and most of these water sources are fecally contaminated. Salmonella is notoriously difficult to culture from the environment, yet we recover it from wells in Ibadan. Reading the genomes of bacteria isolated from different household water sources, we've identified indistinguishable fecally derived bacteria from multiple nearby wells, pointing to the underground contamination and refuting the supposition that Africa's water access problems will be solved by indiscriminately digging more and more wells. Permit me, Mr. Vice-Chancellor, before stepping away from the Trenchard Hall podium, to highlight some of the more pertinent and urgent recommendations that come from a decade of reading genomes in Ibadan, starting with recommendations for water, sanitation, and hygiene. It's no coincidence that the countries where access to improved sanitation is difficult have the highest death rates from diarrheal disease. It's a shame that Nigeria tops those lists, and that people in our great country lose their lives for want of toilets and taps. We must unrelentingly call for simple solutions that eliminated fecal-oral disease in the West over a century ago, even if the infrastructure required to implement them takes longer to build than an election cycle. Mr. Vice-Chancellor, ladies and gentlemen, November 18 to 24 is the 2024 World Antimicrobial Resistance Awareness Week, and it has been my pleasure to share dimensions of this problem with today's audience. We are all antimicrobial resistance stakeholders. Antimicrobial misuse and overuse has fueled a global crisis that requires prompt and decisive action. I want to re-articulate recent recommendations made by the Lancet AMR series, which were presented and adopted by the UN General Assembly in September this year. To get to the 10, 20, 30 by 2030 targets, Nigeria needs to improve infection prevention and control in health facilities, extend vaccines to the unreached, and deliver effective and permanent solutions to our horrific state of water, sanitation, and hygiene. Our work on typhoid, done in collaboration with many excellent scientists and clinicians here in Ibadan, provides more evidence than is needed to support a typhoid application to Gavi. We must devote the necessary resources and energy to measure antimicrobial resistance. Currently, many African countries lack a surveillance system, need support to launch one, and look to Nigeria as a model. I'm immensely proud of my contributions towards starting Nigeria's surveillance system. However, the system needs to grow in size, granularity, and accountability to effectively measure what we need to know about antimicrobial resistance. And it is run entirely by volunteers, and that needs to change if we're to ensure sustainability. The discoveries I shared with you today were made here in UI, but they were supported with grant resources from abroad. It is good that international agencies and philanthropists will invest in our work, but it would be far better if we did so too. As we advocate for Nigeria to devote the 1% of GDP it promised to research and development, may I also plead that non-monetary support for research by our country and institutions be offered as well. The financial, logistic, and consular red tape that hinders rapid procurement, servicing of research and educational materials can and should be cut to foster our country's growth. Mr. Vice-Chancellor, ladies and gentlemen, we may see most of the aforelisted recommendations as big ones targeted at policymakers, most of whom are not in this room and are mere mythical figures in Nigeria. But I have a few other ivory tower recommendations. After all, charity begins at home. Supporting research activity must begin from the ground up. Many younger colleagues turn away from the exacting but rewarding task of applying for grants and building collaborations because there are few incentives to do so. As long as our appointment and promotion criteria devalue large collaborations and give no points for winning competitive grants, colleagues will naturally aim to perform and publish smaller, less impactful studies. Insisting that first authorships weigh more heavily for promotions of biological scientists takes those authorship places away from those most needed by early career scientists, our students. It also means that our postgraduate supervisors, who outside of Nigeria prefer to be listed last on papers, cannot competitively showcase their mentoring on research grants, and consequently, their own publication records are less competitive for research leadership awards. Nigeria's research institutions can do better to launch and support excellence in research, and UI, the first and best, must lead the way. Our research management offices and bursaries must receive stronger support to effectively facilitate grant-winning, execution of grant-funded projects, and good financial grant practice. This, in turn, will bring income and prestige to our university, ultimately making these offices self-funding. I learned to do microbiological science during the excellent master's and doctoral training I had at OAU. But I learned to run a research group, to manage and mentor early career scientists, to write comprehensive review papers, to seek and steward extramural funding as a postdoctoral fellow under the supervision of a research leader. When I completed postdoctoral training, I was ready on all fronts, not only to do research, but to lead it. I strongly suggest that our universities and the Nigerian University Commission normalize postdoctoral scholarship in our academy to improve our research programs and strengthen the pool of future faculty recruits. What recommendations do I have for our talented and ambitious students? Bioinformatics and other computational sciences do not need expensive consumables and can be learned and executed with no more equipment than computers. Currently, undergraduate and postgraduate students taught from our departments now receive some bioinformatics instruction. I thank those of our trainees who are voluntarily cascading the knowledge they have received to peers, and I'm grateful to our faculty's alumni led by Professor S.I. Idowu, who supported a digital innovation studio to make computational biology training possible. 2 generations ago, genomic science did not exist, and all present-day leaders in the field were self-trained. Therefore, Ibadan students, first and best, please work yourselves to learn computational biology. There are tremendously valuable resources and supportive communities online. Computational sciences will be the levelers of global biomedical science progress, which currently lean sharply northwards and westwards. It's hard work, but honest toil to learn Artemis command line, Python, R, or to turn FASTA files into knowledge. Take the challenge, teach yourself, and win the greatness. Become fluent in the language of DNA that is spoken globally by all organisms, including your own cells. Join those of us that read and make sense of biology and contribute to cutting-edge science from your future workplaces or from your hostels right now. It will earn you a great living and it will give you the wherewithal to serve. If you find these or other ways to lead in discovery and health protection, it will matter less which country you live in, what visas you can secure, or whether your government has your interests in view. As one national anthem recommends, build your land's borders inside your heart. I must thank just a few people that contributed to the stories that I've told you today. If you agree that the text of the lecture is admirably proofed, please join me in my gratitude to Ersha Mamoko. Should you stumble across any errors, those are mine. The lecture owes so much to the tremendous amount of selfless effort committed by a faculty officer, Mrs. Chinyere Alawode, and other administrative staff in the Faculty of Pharmacy, the Faculty Inaugural Lecture Committee, led by Dr. Ayorinde, and both my academic departments, Pharmaceutical Micro and Biotech, and Med Micro and Parasitology. I alone could not have bumbled through the bureaucratic, logistical, and technical hurdles that would otherwise have stood in the way of delivering this lecture. Thank you. The work I presented to you today is generously supported by a range of funders and institutions. I'm grateful that they find our work here in Nigeria worth the investment. I've been trained by the best teachers at the best institutions, and I'm grateful for all that was invested in my intellectual development. My mentors steered carefully but distantly, ensuring that I could explore without dropping off cliffs, and showing me in turn how best to support my students. Preeminent among them are Professor Adebayo Lamikora, who I'm so honored is here, who supervised my undergraduate, master's, and doctoral dissertations, and Professor Jim Caper, who supervised my PhD and postdoc. They continue to mentor to this day. I was prepared by and nominated for most of the honors I received by at least one of them. And I've not been successful by coincidence, nor am I an outlier. The peers I'm most proud of were their mentees as well. I'm grateful to Professor Chinedum Babalola, who, when I was searching for a new academic home in Nigeria, invited me to UI and showed by her own accomplishments. She showed by her own accomplishments that it's possible to do high-quality, impactful, extramural supported work in the Faculty of Pharmacy. I thank successive deans, provosts of the College of Medicine and Postgraduate College, Deputy Vice-Chancellor, and Vice-Chancellors. I'm grateful to the College of Medicine for smoothing the way for fruitful collaborations, permitting me to domicile my research and broadcasting this lecture so that it can be heard online. I particularly want to thank the immediate past Provost of the College of Medicine, Professor Migbodu, for going over and above these commitments to inviting me to join the college faculty as an adjunct, and CRIM coordinators and staff working with us on GFGP certification. I've worked with amazingly talented collaborators. Almost 120 of them were students when they worked with me. I have endeavored to mention key student discoverers in the course of my talk. I'm thankful to the rest of you who I was not able to discuss your projects. Postdoctoral researchers, Drs. Afolayo, Androga, and Ikumuko, accomplished great things with us, whilst I mostly stay out of their way. My lab is a vibrant place populated by promising young scientists. In spite of the uncertainty that comes with discovery in Nigeria, we only manage to circumvent what others call risk, because students and staff work well supported behind the scenes by a resourceful and resilient administrative team of ours. Thank you all. I thank them and the present staff of SETA, ACON, and TESS at the College of Medicine. I belong to an excellent department of pharmaceutical microbiologists and biotechnologists, and we work within the Faculty of Pharmacy that is a stimulating academic home for our work. My second department, medical microbiology and parasitology, contributed much heft towards supporting this lecture and serves as an important home for the clinical bacteriology research that we do. Further afield, I'm grateful that for the 2 decades I've collaborated with Professor Oladipo Abodering from IFE and our productive alliance with NCDC. And through our interactions with the Africa Center for Disease Control, my group members and I have enjoyed the opportunity to learn about antimicrobial resistance and genomic work across the continent and to influence policy. Over the course of my career, I've relied on professional associations and built a network of collaborators. Thank you, all of you. I think OWSD is here. Thank you. I enjoy an idyllic life at the University of Ibadan, and I'm grateful to the entire community for welcoming our family a few years ago and making us comfortable enough to call it home. I especially want to thank the chaplaincy, congregation, women's fellowship, and guild of ushers of the Chapel of the Resurrection. I also thank University Health Services for their care over us, our students, and our guests over the years. And thanks to residents on my streets, on campus. All of you make us home. I thank my parents, Engineer Obed and Mrs. Flora Okeke, for working around the war and structural adjustment to bring 4 children born in exile to be schooled back home, making it possible for me to conceive a life and career in my own country. My mother, herself an alumna of UCH's nursing school, recognizes that women can do everything, and therefore her daughters could dream of careers in engineering and science. I thank my parents for sending both my sister and I to Queen's College, Lagos, where we will be able to light and pass on torches and gain a network of lifelong friends and supporters who have achieved more impressive feats than we could ever imagine. My uncles, Samson and Chukwuma, Ezemokolo, and their families, delighted to have scientific nieces, made it possible for my siblings and I to continue to live and train here in Nigeria with or without residence parents, and so became our nuclear family as well. I wish to thank the entire Okonta family, including those recently departed, for welcoming and cheering a very late bride. While none of my siblings can be here at the time, Chiji, Uchenna, and Ebele are the greatest champions of my unusual academic path and Jakpada adventure. They are also 3 of my best friends. One more is dearest Dr. Ike Okonta, who believes that someone has to make Nigeria's future and is bold enough to support my own feeble attempts in this regard. And of course, we both thank our friend and daughter, Miss Chika Okonta, who joined the journey we mapped out before she was born, but gets it in extraordinary ways. May I end by thanking and giving all glory to God, who makes things that make themselves and gives us so much pleasure from reading scripts written in organisms' genomes.